Welcome to this explainer. I'm absolutely thrilled to have you here today. We are about to embark on an epic journey, diving straight into the microscopic world. It's a realm completely invisible to the naked eye, yet, think about this, it literally runs the operating system of our entire planet. We're going to explore the strict rules of cellular life, and then we're going to meet the ultimate biological hackers that just shatter every single one of those rules. Let's get right into it. So, what actually separates a living cell from a microscopic hijacker? Honestly, it's a profound mystery. On one side, you've got complex, self-sustaining microscopic life. On the other, tiny, stripped-down biological machines that literally can't even reproduce without stealing someone else's factory. The contrast is just incredible. And understanding this fine line between the living and the not quite living, it's a total game changer for how you view biology entirely.
Okay, let's dive into this. Here's our roadmap for today. We'll start with understanding bacteria, then look at bacterial superpowers, compare prokaryotes versus eukaryotes, before moving on to the viral invaders, and finally, how viruses survive.
Section 1. Understanding bacteria, the rule makers.
It all starts right here with the prokaryote. A prokaryote is basically a unicellular organism that completely lacks a nucleus or any membrane-bound organelles. Think of it as life stripped down to its absolute most foundational architecture. Now, it's really crucial to note that both archaea and bacteria fall under this massive biological umbrella. They are, for all intents and purposes, the original rule makers of life on Earth.
So what actually comes standard when you're a bacterium? You can kind of think of this as the basic starter pack for cellular life. Every bacterial cell relies on this standard equipment to function. First up, you've got a tough outer armor, a cell wall made of peptidoglycan, which is also known as murine, and that's purely for protection. Just inside that is the cell membrane, keeping everything contained within a fluid called cytoplasm. Now, instead of a neat, packaged nucleus, their genetic blueprint is just this loose string of circular DNA, and that hangs out in a region called the nucleoid. And finally, they've got ribosomes, which are essentially these tiny cellular factories that synthesize proteins.
And this brilliantly illustrates biology in its purest form, right from our foundational source notes. They map out the raw structural blueprints, like that lack of a double membrane nucleus we just talked about, in the presence of that peptidoglycan cell wall. These are the fundamental rules that dictate exactly how these organisms operate.
Moving right along to Section 2, Bacterial Superpowers, Optional Upgrades.
Now, not all bacteria are created equal. Some actually come with these absolutely incredible optional upgrades. They might sport a capsule, that's a thick polysaccharide shield protecting them from drying out, or it even stops antibiotics in their tracks. Then you have plasmids. These are quite literally biological cheat codes. They're small circular pieces of DNA that bacteria can trade with each other to grant things like antibiotic resistance. And how do they trade them? Well, they use pili. These are protein poles that vary in length and stiffness, and bacteria use them to physically connect and swap those genes. Some also have flagella for swimming, and others have membrane infoldings. Those basically act as extra surface area for some serious biochemical heavy lifting, like photosynthesis or nitrogen fixation.
Delving into the source notes here, I really want to highlight a highly specific and honestly fascinating mechanism. Check out the details on the flagellum. It's actually not just some loose whip that thrashes around aimlessly. The notes specify it's a rigid, hollow cylinder made entirely of identical protein molecules, and it works by rotating right at its base, spinning in a corkscrew-shaped motion, literally just like a tiny biological outboard motor propeller pushing the bacteria through its environment. Talk about a masterpiece of microengineering.
Section 3, Prokaryotes versus Eukaryotes, Scaling Up Life.
Now, what's really interesting about this slide is the sheer scale of the upgrade from prokaryotes to eukaryotes. Just look at the diameter row. Prokaryotes sit around 1 to 5 micrometers, but eukaryotes, they scale way up to 40 micrometers. And because we exist in 3 dimensions, well, that actually means eukaryotes have up to 1,000 times the volume of a prokaryotic cell. That's massive. We also see a major shift in how they reproduce. While simple bacteria just split right down the middle via binary fission, eukaryotes develop the much more complex processes of mitosis and meiosis. The genetic material gets locked safely away inside a nucleus, and the whole shebang just becomes vastly more intricate.
But, scaling up comes with some pretty significant trade-offs. When we compare their abilities side by side, eukaryotes evolved complex cell walls made of cellulose in plants or chitin in fungi, but, and this is a big but, despite lacking membrane-bound organelles, those simple prokaryotes retained a unique superpower that complex eukaryotes lost completely. I'm talking about nitrogen fixation. No complex eukaryotic organism on Earth can pull nitrogen directly out of the air. No way. We literally rely entirely on the simpler prokaryotes to do that heavy lifting for the entire global ecosystem.
Let's move along and see how this builds, because as we scan further down the source text, we finish comparing these two majestic forms of cellular life and immediately run into something entirely different. Just a tiny, stark heading that says, virus. We are officially leaving the established rules behind, folks, and entering the dark web of microbiology.
Section 4. The viral invaders, the rule breakers.
The notes define them with chilling simplicity. Very small, infectious particles. That's it. They don't have standard equipment, they don't have cytoplasm, they don't even have their own ribosomes to make proteins. They are, quite simply, a rogue genetic payload wrapped up and actively looking for a target. To truly grasp just how rogue they are, you really have to understand the scale here. 20 to 300 nanometers. Remember earlier when we said a bacterium was 1 to 5 micrometers? Well, a micrometer is 1,000 nanometers. So, to put that into perspective, this means a virus is just a tiny, tiny fraction of the size of the bacteria we were just looking at. They are vanishingly small. This makes them incredibly difficult to detect and absolutely perfectly sized to infiltrate living cells.
Their anatomy is honestly terrifyingly efficient. They typically have this symmetrical shape that contains nothing more than a self-replicating molecule of either DNA or RNA. This payload is protected by a protein coat called a capsid, and that's built out of individual protein molecules called capsomeres. Now, some viruses take it one step further. They wrap themselves in a phospholipid envelope, which, if you think about it, is really just a membrane-like outer layer of lipids and phosphates that they've straight up stolen from the host cells they previously destroyed.
Section 5. How viruses survive. The ultimate hackers.
So the crucial point is captured perfectly in this single quote. All viruses are parasitic because they only reproduce by infecting or taking over living cells. Think about it. A virus sitting on a table does absolutely nothing. None. It has zero metabolism. It cannot eat, it cannot grow, and it definitely cannot multiply on its own. It is entirely 100% inert, right up until it makes contact with a vulnerable host.
The viral life cycle is a literal hijacking of the cellular machinery we learned about earlier. Step 1, they infect a living cell, breaching its outer defenses. Step 2, their viral DNA or RNA completely takes over the cell's internal factory. You know those ribosomes we talked about earlier? Yeah, the virus hijacks them. It forces the host cell's protein synthesizing machinery to completely stop doing its normal, healthy job and instead read the viral genetic code. And then step 3, the poor cell is forced to manufacture hundreds, sometimes thousands of brand new virus particles. This usually destroys the host cell in the process, as the new army bursts out to infect others. The idea spreads like wildfire, basically.
Which leaves us with one of the most hotly debated questions in all of biology. If viruses completely lack the fundamental standard equipment of life, if they have absolutely no metabolism of their own, and if they can only ever replicate by hijacking the machinery of actual living cells, are they truly alive? Or are they just incredibly complex, rogue chemical algorithms? I'll let you ponder that as you digest today's breakdown. Thank you so much for joining me on this explainer. Keep questioning the invisible world around you, and I'll see you next time.